Hi everyone, my name is Faeze and I'm a PhD candidate at University of British Columbia. Today I will be presenting my poster titled Machine Learning Derived Transcriptional Signatures in Cancer. As all of you know, modifications in DNA molecules can alter protein functions and cellular pathways they participate in. Of course, these modifications in cellular pathways can lead to cancer, and it is crucial to study the impact of mutations on cellular pathways to identify tumor vulnerabilities. Transcriptome, which is the set of all RNA molecules in cells, can help us gain a better understanding of cellular modifications in cancer. Thanks to the wealth of omics data generated through next-generation sequencing technology, now we have the opportunity to use machine learning approaches to investigate the impact of mutations on the transcriptome. So to study the impact of TP53 gene mutations on the transcriptome, I used two datasets. The first one is the Cancer Genome Atlas, or TCGA, that contained 8,755 primary tumor samples, and closer to 40% of them had a mutation in TP53. I divided the samples in this group into two categories based on the impact of mutations, which is based on the variant consequences from the Ensemble database, and we only used the samples with a high or moderate impact mutation during training phase. The second set is from the BC Cancer Personalized Oncogenomics Program, or POC. In this set, we have 570 metastatic tumor samples, and closer to 50% of them had a mutation in TP53, and I further also divided these based on the impact of mutations. So the reason I decided to start this analysis with TP53 is due to its tumor suppressor activities, and for that reason, it is called the guardian of the genome. It has a high mutation frequency in cancer, and about 5 to 80% of samples have a mutation in this gene depending on tumor type and stage. So using a random forest model, I divided the samples based on mutational status of TP53, and I observed that the classification can be performed successfully in both TCGA set of primary samples and POG set of metastatic samples. And even after we merged the two sets, not only the classification could be performed really well, but also we observed some improvements. To make sure that the good performance of the model was not just based on one or two tumor types, I divided the samples based on their respective tumor types. And here on this graph, the y-axis is the F1 score of classification, and the x-axis shows the ratio of minor to major class size. Here, minor or major class can either be the set of samples with mutant copies of TP53 or the set of samples with wild-type copies. And the reason that these minor and major classes can be different for each tumor type is because we do have different rate of mutation in this gene in different tumor types. And as you can see in this graph, for majority of tumor types, the samples could be classified really well based on mutational status of TP53. And thus we concluded that the transcriptional signature of this gene is universal across cancers. In the next step, I extracted the genes that had a high importance scores based on the model, and we identified a lot of targets and regulators of TP53, and also a lot of the genes had some association with this gene. And this was a further proof for us that the algorithm not only could classify the samples really well, but it could also learn something about TP53 biology. So of course, naturally, the next step was to expand the analysis to other cancer genes. And for that reason, I selected 50 genes either based on their mutation frequency or their known role in cancer initiation, progression, or maintenance, and divided the samples or classified the samples based on mutational status of these genes. And as you can see on the y-axis, we have the F1 score of classification. And on the x-axis, we have the ratio of mutant to wild-type samples. As expected, for majority of these genes, the number of samples with a mutation is really small compared to the samples containing wild-type copies of these genes. However, we can still see that the algorithm can perform really well for some of these genes. And more specifically, I'm going to talk about BRAF gene here. So similar to TP53, I divided the samples based on their respective tumor types. And as we can observe, here only the performance was good in one or maybe two tumor types, and this can suggest that the transcriptional signature of BRAF is more tumor type specific. So in terms of future directions, I'm planning to analyze the performance of the algorithm on all the selected genes across different tumor types and compare the performance between different sets of tumor samples to evaluate if the performance is more tumor type specific or universal across cancer types. I'm also planning to perform multi-label classification for genes that belong to the same pathway to understand the impact of pathway interruptions. Finally, I'm planning to employ the genes and pathways that we found in this analysis to discover new molecular targets for therapy. Finally, I want to thank all the patients and their families who participated in these studies, my supervisor, Dr. Stephen Jones, Dr. Erin Pleasant for all her help throughout the project, all my funding sources, and thank you all for listening.